All right, we are going to start with number 9 now. So it says the diagram above represents a typical rod-shaped bacterium. Bacterium, as you might remember, is prokaryotic. So this is a proc, meaning no nucleus and no membrane-bound organelles. Which of the following best describes a feature shown in the diagram that is unique to archaea and bacteria? We didn't talk about archaea a lot, but that is also a prokaryote. So it says the organism is surrounded by a cell wall. Well, that is not unique, right? Plants also have cell walls. Insects have exoskeletons that have cell walls. So it's not that one. The organism contains ribosomes. Again, that's a true statement. However, everything has ribosomes, so that is not unique. The organism does not have a nuclear membrane surrounding its genetic material. So it's saying it doesn't have a nucleus that houses its DNA or RNA. That's looking pretty good because we just said it has no nucleus. So that's probably right. D, the organism is not capable of making and providing itself with ATP. Well, everything needs ATP energy in order to survive, so it is in fact C. Number 10, we have 2 graphs here. So I'm going to read the question before I get into the graphs. It says all the following changes would be likely to decrease the rate of transpiration at 8 a.m. except. So we're looking for one that decreases the rate of whatever that is at 8 a.m. Here's 8 a.m., here's 8 a.m., just for reference. Now, I need to know what the rate of transpiration is. It tells us it's the flow of water through the stem and leaf. Water potential are measured in a tree during a 24-hour period under normal environmental conditions. So here's our tree or a plant or whatever. Transpiration is the act of the water going from there, out to the environment. Okay, right? The flow of water through the stem and leaf water potential are measured. All right, so which of the following will decrease the rate of transpiration? Well, here we have leaf water potential and transpiration and stem flow. So causing the stomata to close, would that decrease the rate of transpiration? Maybe. Because if we're closing this, then maybe the water can't get into the environment. So that's a maybe. B, increasing the humidity of the atmosphere. So if I increase humidity, so we have a lot of water in the environment, then we're going to have a lot more water being able to be taken up through the plant or the tree. Increasing the water potential of the atmosphere. So if I increase the water potential of the atmosphere, that means it would be less likely for it to leave the tree to go to the atmosphere. And so that's going to actually potentially decrease the rate of transpiration. Let's see. All the following changes would be likely to decrease the rate of transpiration. So yeah, this one looks like it would decrease it, right? This one, the wording is tricky, right? It's all the following charges would be likely to decrease except. So the right answer will actually increase the rate of transpiration. So just causing the stomata to close, that should decrease it. Increasing the humidity of the atmosphere would likely decrease it, but I'm not so sure about that one. Increasing the water potential of the soil. Well, if it's going from the roots up through the stem, the leaves, into the atmosphere, it looks like increasing the water potential of the soil would allow this process to happen faster and increase the process. So it looks like D is the correct answer here. Let's think about B, because I was unsure about that. If we increase the humidity of the atmosphere, that means there's less of a potential difference between the leaves in the atmosphere, and so the rate of transpiration out of the plant is going to be slower. So it is, in fact, D. Now this one starts here, and then we're doing questions 11 through 13 based on all this background info. So number 11 says, which of the following graphs is most appropriate representation of the experimental data? So if I go back here, and I'm looking at what the heck they're looking at, I have a treatment group, 1, 2, 3, 4, 5, and in these groups they have these different solutions given, and we are measuring the mean absorbance of light. Okay, so if the groups are 1, 2, 3, 4, and 5, you can't have a group 1.5, you can't have a group 4.8. These are just different discrete categories, so this is going to be a bar graph. So I'm looking here. Which one's a bar graph? Well, it's either this one or this one. This one has error bars. This one does not. This one we'll have to see if the numbers are more accurate or if these are more accurate. So looking back here, let's look at 1 and 2. 1 is 0.16, 2 is 0.12, so 2 should be lower than 1. This one has 2 lower than 1. This one has 1 or 2 greater than 1. So it's not going to be this one, it is going to be A. Plus this one has error bars, and we are given the 2 times the standard error, so we are given error bars. All right, so that one is pretty easy. You just needed to know it was a bar graph. So now 12 says, which of the following is the dependent variable in this experiment? So the dependent variable is going to be your mean absorbance of light, because that is what is being measured. Yep, so it would be D. Don't have to look into that too much. It's just whatever's on the y-axis. 13, based on the data from the investigation, which of the following is the best scientific question about organisms living in water that is polluted with organic solvents? So now I have to actually read the background information to figure out what the heck is going on. So it says, a student formulated a hypothesis that water-soluble pollutants, so pollutants that can be broken down with water, damage living organisms by increasing the permeability of cell membranes. So this is the claim, right? So we're basically saying that these pollutants are taking a cell membrane and making it more open to the environment. All right? To test the hypothesis, the student investigated the effect of isopropanol and acetone on beetroot cells. So I don't really need to know what those are, but they're some sort of chemical. The vacuoles of beetroot cells contain large amounts of beta-cyanine, a water-soluble pigment that is released into the extracellular environment, so outside of the cell, as a result of increased membrane permeability. All right? So if I have more permeability, I have more beta-cyanine in the environment. The student prepared identical samples of beetroot tissue and incubated each sample for 15 minutes in the specific solution for that group, which is given here. At the end of the incubation period, the student measured the absorbance of that amount of light for each sample. A greater concentration of beta-cyanine in the solution surrounding the beetroot results in a greater absorbance of light. So increase light absorbance, we have increased beta-cyanine, and if we have increased beta-cyanine, then that supports the hypothesis, right? This is basically saying that then there is more permeability of the cell membranes. So now, based on the data, which of the following is the best scientific question about organisms living in water that is polluted with organic solvents? Do organisms without pigments have a selective advantage in polluted environments? We haven't talked specifically about selective advantage yet, but it seems like an advantage, something that would help them in polluted environments. I'm not sure we can answer this question based on the data that is given, so we'll come back to that. Will organisms living in polluted environments exhibit detrimental effects from an increased permeability of their cell membranes? Well, this is what we were looking at, right? If they had increased permeability of the cell membranes, and it was caused, allegedly, by the polluted environment, so this one is looking pretty good. C, will organic solvents from the environment become incorporated into the cell membranes of organisms? We are not studying that, right? We are just seeing if there's more light absorption, therefore more beta-myosin, or whatever, and then therefore more membrane permeability. So no. D, should governments place more stringent restrictions on the release of organic solvents into aquatic ecosystems? That is not where we're getting to yet. That might be a discussion question for this investigation of using it to support that. However, the best question about organisms living in water is going to be B. Will it have detrimental effects from an increased permeability? All right. Number 14. All right, lots of background. Let's get to the question. And this one is specifically for questions 14 through 16. So it says, based on the data shown, changes in the light intensity resulted in changes in the rate of which of the following processes? So I'm going to have to read the background info. I'll let you read it with me. So we're studying light intensity on producing oxygen in green algae, oxygen production. So this is going to be photosynthesis. The algae were, because that is a product of photosynthesis, the algae were suspended in water inside a sealed glass jar, and the jar was placed into a constant temperature, light-proof box containing a light source. A probe was inserted into the jar to record the concentration of oxygen. The probe was connected to a recording device. The setup is like this. The student decreased the intensity of the light hourly. So over time, we're decreasing the amount of light, and recorded the corresponding changes in oxygen concentration. We can see it decreased oxygen concentration. Okay, changes in light intensity resulted in changes in which of the following processes? This is going to be photosynthesis, like we said, because that's a product of photosynthesis is oxygen. So now 15 says, the rate of oxygen production equaled the rate of oxygen consumption during which of the following time periods? Okay, so if I'm looking, the rate of oxygen consumption, so this over time, is equal to the rate of oxygen production. It's going to be when those time periods are equal. So from J to K is the only one where it's actually equal. And so it is going to be D. D as in dog. Number 16. An increase in the rate of oxygen production by algae would be accompanied by a comparable increase in the rate of production by which of the following substances? So if I'm increasing oxygen, what else is a byproduct of photosynthesis? Well, that's glucose, right? We're making glucose. Carbon dioxide is going in. CH4 is methane, that's not involved at all. And water is also going into a plant, so it is A. 17, it says, the figures below illustrate the similarities between ATP synthesis in mitochondria and chloroplast. Remember, mitochondrion is one mitochondria. Mitochondria is the plural. The figures can best assist in answering which of the following questions. Do electron transport chains create a gradient so that ATP synthase can generate ATP molecules? Well, here we're seeing these different molecules making a gradient between a membrane that then goes on to make ATP. So honestly, A is looking pretty good right now. B, what are the sources of energy that drive mitochondrial and chloroplast electron transport systems? So we have NADH coming in here, It doesn't show anything about light over here. It just sees water splitting to replenish the electrons. So that one is not looking good because it's not saying like light energy is coming in or NADH is providing energy to these proton pumps. C, what is the optimal temperature? Blah, blah, blah. Does this say anything about temperature? No, so it is not C. D, what is the evolutionary relationship between ATP synthase and mitochondria and ATP synthase in chloroplast? Looking here, it's not talking about how it changes over time or how they're similar. It's just showing how proton gradients are being formed. So it is, in fact, A. We'll stop there and make another video.